Hi, and welcome to this series exploring landmark experiments in protein science based on my book of the same name, which can be purchased on Amazon, and that link is in the description. In the video on genetics, I talked about how chromosomes in the nucleus were suspected of containing the units of heredity called genes, but no one really knew what they were. In this video, I'll expand on this topic and describe the discovery of nucleic acids and the elucidation of the structure of DNA. In the late 1860s, Friedrich Miescher was isolating proteins to determine the chemical composition of cells and found a material which was most likely to be attributed to cell nuclei since it was more acidic than proteins and which precipitated in acidic solutions and then redissolved in an alkaline solution. So to get larger quantities of material in order to study its composition, he went to a nearby surgical clinic and collected freshly used bandages since they contained lots of white blood cells. After making sure that the samples had not started to decompose, he washed the bandages with sodium sulfate since, contrary to other solutions, it didn't make the cells swell to a slimy consistency, and then he filtered out the cotton fibers. Miescher then allowed the cells to settle to the bottom of a tube for a few hours, so remember from last video that Svedberg had not invented his efficient centrifuge yet, and from the settled cells he poured out the supernatant to isolate just the cells. And so to isolate the nuclei from these cells, Miescher first tried dissolving the protoplasm in dilute hydrochloric acid for several days, but this didn't quite work out since in the majority of cases some of the protoplasm stubbornly adhered to the nuclei. But after treating the cells with an acidic solution for several weeks in the cold of winter, meaning at low temperatures, so like the centrifuge, refrigerators had not been invented yet, so he literally had to perform this experiment in wintertime. But anyway, after treating the cells in this way, he was able to isolate pretty clean nuclei, which he confirmed by a lack of staining with an iodine solution, which stains proteins. Miescher then shook the solution vigorously in ether and water to separate nuclei that still had protoplasm attached and which collected at the interface between the two liquids, whereas naked nuclei, which were those that were completely clear of protoplasm, settled as a fine powdery sediment at the bottom of the tube. Miescher then used dilute sodium carbonate to extract from the nuclei a yellowish solution of a substance that, when treated with dilute acetic acid or hydrochloric acid, yields a flocculant precipitate insoluble in an excess of acid. And this precipitate was the first purification of DNA. And since the substance was isolated from nuclei, he called it nuclein, but it was later renamed nucleic acid. He also thought that it was likely that a whole family of such phosphorus-containing substances, differing somewhat from each other, will emerge as a group of nuclein substances, which perhaps will deserve equal consideration with the proteins. I cannot help thinking that here lies the most essential physiological role of phosphorus in the organism. A knowledge of the relationships between nuclear materials, proteins, and their immediate products of metabolism will gradually help to raise the curtain, which at present so completely veils the inner process of cellular growth. The discovery of the building blocks of nucleic acids, called nucleotides, is more complicated than that of proteins since they readily break down into nucleobases, also called nitrogenous bases since they contain a lot of nitrogen, when treated with acids. Guanine was first isolated from bird excrement, which is called guano and hence the name, in 1844, but it was only confirmed to be part of nucleic acids in 1879 when it was isolated from it by acid hydrolysis, and adenine was similarly isolated from beef pancreas nucleic acid in 1885. These two molecules are part of a family of molecules known as purines, so named by Fischer in 1884 from the word purum, pure, and uricum, urine, since the first purine molecule was isolated from urine. These molecules are composed of one 5-sided imidazole ring and one 6-sided pyrimidine ring, and the difference between the two molecules is the placement of the nitrogen. Thymine and cytosine were discovered in 1893 and 94, respectively, from calf thymus and beef spleen, and uracil was first synthesized in 1884 and subsequently isolated from nucleic acids early in the 1900s. Pinner proposed the ring structure of these molecules and called them pyrimidine in 1885, a portmanteau of the word pyridine, which is the ring structure, and amidine, the segment containing the two amino groups and the oxygen between them. Walter Jones commented in 1914 that the discovery of these molecules was considered to be of great significance since it gave character to the nucleic acid and furnished a definition of the substance by which it could be distinguished from proteins and other constituents of the body. It made possible a study of the distribution of nucleic acid in the body without actually separating the cell nucleus from the protoplasm. It furnished a method of distinguishing true nucleons from pseudonucleons. It furnished the refutation of the persistent claims that nucleons are merely compounds of proteins with metaphosphoric acid. It suggested in no uncertain way a chemical connection between the cell nucleus and urinary uric acid. As already mentioned, isolation of these molecules simply came as a result of the complete breakdown of nucleic acids using strong reagents. However, determining the building blocks of nucleic acid would not be so easy because gentler methods would need to be optimized. And although many different purines and pyrimidines exist in cells, only thymine, uracil, adenine, guanine, and cytosine make up the nitrogenous bases of nucleic acid. In addition, a number of disturbing factors have continually operated to confuse experimental findings. Investigations have been conducted with impure material at a time when dependable methods of separating decomposition products were not available. Products of secondary decomposition were assumed to be primary, and substances formed from adherent impurities have been ascribed to the nucleic acids themselves. In 1909, Levine and Jacobs showed that inosinic acid could produce two different compounds depending on the type of hydrolysis used. Using acid hydrolysis resulted in a compound composed of phosphoric acid and a pentose sugar, whereas hydrolysis with a neutral reagent yielded a compound composed of pentose sugar and hypoxanthine, which is a breakdown product of adenine. This was the first purification of these types of molecules, and the authors called the purine carbohydrate compound nucleoside and the phosphate ester of a nucleoside, such as inosinic acid, a nucleotide. Therefore, these and other experiments showed that nucleotides were composed of phosphoric acid linked to a pentose sugar, which in turn was linked to a nitrogenous base, either a purine or a pyrimidine, depending on the nucleotide. However, in contrast to the pentose sugar found in inosinic acid, which had been isolated from meat extracts and was determined to be ribose, another pentose sugar, deoxyribose, had been isolated from other animal nucleic acids, and further still, both ribose and deoxyribose had been isolated from yeast, and this led to considerable confusion. Eventually, yeast and plant nucleic acids were thought to contain the three common bases along with uracil and the sugar ribose, and so they were called the ribonucleic acids, or RNA, whereas animal nucleic acids were thought to contain the three common bases and thymine, as well as deoxyribose, and so these were called deoxyribonucleic acid, or DNA. Both types of nucleic acid contained phosphoric acid. However, in 1924, Foilgun and Rosenbeck made use of a DNA stain, now called the Foilgun stain, to show that DNA was present in the nucleus of all cells, regardless of their origin. In contrast, RNA was shown to be mainly located in the cytoplasm, although some was also found in the nucleus, and it became clear that both nucleic acids were found in all cells. However, the difference between these molecules, as well as how their nucleotides were linked, was yet to be determined, but preliminary data suggested that DNA was linked to cell division, whereas RNA was linked to protein synthesis, since it was found to be present in large amounts in cells where protein synthesis was abundant. The role of RNA in protein synthesis will be discussed in my video on protein synthesis. The first significant step towards showing that nucleic acids were responsible for heredity came in the 1920s when Fred Griffith noticed a 5-year trend where there was a significant decrease in the incidence of type 2 and a corresponding increase in type 4 pneumococci in pneumonia patients. He also reported a particularly striking occurrence where a patient had 1 type 1 and 3 type 4 pneumococci at the same time. To investigate this, and in the course of other inquiries and of routine examination of sputum from pneumonia patients, he introduced samples of patients' bacteria into mice, and, not surprisingly, these mice died within a few days. But when he examined what type of bacteria the mice had when they died, he found that some mice that were injected with one type of bacteria harbored another type. And so to explain these experiments a bit better, Virulence in pneumococci can easily be distinguished by two different phenotypes. Smooth bacteria, the S form, are virulent and their colonies have a smooth appearance, whereas rough bacteria, the R form, are not virulent and their colonies have a rough appearance. But Griffith found a mouse which died from a rough bacteria, which possessed a combination of rough cultural characteristics and virulence which had not previously been noted. So he suggested two hypotheses for this. One, either the less invasive strains, the type 3 and 4, were already present in the mouse when the more virulent strains, types 1 and 2, were injected, or two, the inoculated strain somehow transformed into another strain via an unknown process, but Griffith admitted that, on a balance of probabilities, interchangeability of types seems a no more unlikely hypothesis than multiple infections with 4 or 5 different and unalterable serological varieties of pneumococci. And so to test the transformation hypothesis, Griffith carefully set up experiments in which he inoculated mice with one type of bacteria, either the S or the R, along with heat-killed bacteria of the other type. Interestingly, he found that when the S form of bacteria, which is the virulent type, were heat-killed, they could indeed transform the R type into a virulent strain when they were inoculated together in mice. However, he found that the temperature of the heat treatment was critical. Temperatures that were too low or too high would prevent transformation. And so in this column, you have the characteristics of the type of S bacteria that was heat-killed before injection. In this column are the characteristics of the living bacteria that was injected along with the heat-killed bacteria, and these were all R-type. And the results showed how long before the mice died of the infection or was killed to assess the bacterial culture present. Notice how several of the mice had the S-type bacteria even though they were injected with live R-type bacteria. However, identification of this transforming principle would have to wait until 1944, when Avery, Macleod, and McCarty optimized the conditions for transformation and performed a variety of tests to identify the purified substance. They determined that it was not a protein since it didn't have any peptide bonds, but the ratio of nitrogen to phosphorus was within the range that was expected for DNA. In addition, DNase, which is an enzyme that degrades DNA, was found to inactivate the transforming principle. These results were all the more surprising since DNA had not been shown to be present in pneumococci. The authors concluded their paper by stating that if the biologically active substance isolated in highly purified form as the sodium salt of desoxyribonucleic acid actually proves to be the transforming principle, then nucleic acids of this type must be regarded not merely as structurally important but as functionally active in determining the biochemical activities and specific characteristics of pneumococcal cells. Assuming that the sodium desoxyribonucleate and the active principle are one and the same substance, then the transformation described represents a change that is chemically induced and specifically directed by a known chemical compound. If the results of the present study on the chemical nature of the transforming principle are confirmed, then nucleic acids must be regarded as possessing biological specificity, the chemical basis of which is as yet undetermined. Up until the 1940s, the accepted hypothesis about the structure of both deoxyribonucleic and ribonucleic acids was Levine's tetranucleotide theory proposed in 1909, which suggested that nucleic acids were formed by the end-to-end joining of each of the 4 nucleotides in the determined and invariable sequence. However, this theory started to crumble when it was discovered that nucleic acids were much bigger molecules than expected, and in fact they were the biggest known molecule, and this led to the suggestion that tetranucleotides form polytetranucleotides, which consisted of end-to-end joining of tetranucleotides. In hindsight, Gulland admitted that the tetranucleotide theory would never have gained any traction if the true molecular weight of the molecule had been discovered earlier, stating that it should be realized that the conception of a molecule composed of polymerized tetranucleotides has grown from a mental superposition of the later demonstrations of high molecular weights on the older ideas of a simple molecule containing one of each of the 4 appropriate nucleotides. Had the true molecular sizes been realized earlier, it is doubtful whether the conception would have gained such firm hold as is apparently the case. At present, the facts tend to favor the hypothesis of molecules composed of polymerized tetranucleotides in the cases of yeast ribonucleic acid, and some examples, at any rate, of desoxypentose nucleic acids, but sound evidence is non-existent either to support the existence of a uniform tetranucleotide composed of one of each of the appropriate nucleotides, or to indicate a regular sequence of nucleotides. Turning to pentose nucleic acids in general, there are contrary indications to a uniform content of the four nucleotides. It was in the midst of these controversies that Erwin Chargaff proposed in 1944 that the genetically significant differences determined by DNA might be reflected in analytically detectable differences in the content and order of the DNA bases, i.e. the purines and the pyrimidines. And in the introduction of the first of several papers published in his laboratory between 1948 and 52, the authors concluded that it is hard to conceive of two macromolecular substances, that is, DNA and RNA, synthesized in as different cellular systems as cap thymus and yeast, as being identical, although in the absence of immunological or more refined chemical methods, a strict decision may not yet be possible. They went on to describe their improved method for isolation of intact DNA in yeast, which allowed him to quantify the nucleotides present in nucleic acids more precisely. However, Miescher and Chargaff had to find a better way to separate nucleotides using paper chromatography, which is a technique where molecules in solution are separated by passing them through filter paper. So they passed a known mixture of nucleotides dissolved in a variety of solutions through filter paper, dried the paper in an oven, and a mercuric complex of the nucleotides was formed by adding a mercuric nitrate to the purines or mercuric acetate to the pyrimidines. Then, the filter paper was passed through a solution of ammonium sulfide, which resulted in well-defined black spots of mercuric sulfide, which are then cut out and air-dried, after which the DNA was extracted using hydrochloric acid. The amount of nucleotide present in each spot could be quantified by exposure to ultraviolet spectroscopy, since it was already known that DNA absorbs ultraviolet light in a manner that was proportional to its concentration. And here each lane represents a sample prepared in a different solution, and the authors were basically trying to determine which solution was best to separate each nucleotide from one another. And so with this technique in hand, Miescher and Chargaff studied the breakdown of pyrimidines following the harsh treatments that were required to study them. The same basic molecule forms the pyrimidines, cytosine, thymine, and uracil, but differ only by addition of one molecular group, and cytosine was known to be converted to uracil quite easily. So it was possible that the harsh treatment used to study nucleic acids might result in these alterations, and for this reason, the presence of uracil in nucleic acids was put in question by many. Therefore, Miescher and Chargaff used paper chromatography to run pyrimidine samples which had been subjected to a variety of chemical treatments and found that using concentrated formic acid instead of strong mineral acids to extract these nucleotides preserved cytosine and prevented its conversion to uracil. They also investigated the distribution of nitrogenous constituents of pentose nucleic acids in yeast and pig pancreas, and showed that uracil was indeed present in ribose nucleic acids, and it wasn't just converted from cytosine. They also confirmed that thymine was not present in RNA. But most importantly, this study conclusively showed that nucleotides were not present in equal amounts as dictated by Levine's tetranucleotide theory, and set in motion the demise of a paradigm that had existed for nearly half a century. Furthermore, they investigated the composition of DNA in samples obtained from various sources and found that the desoxypentose nucleic acids extracted from different species thus appear to be different substances or mixtures of closely related substances of a composition constant from different organs of the same species and characteristic of the species. It is, however, noteworthy, and whether this is more than accidental cannot yet be said, that in all desoxypentose nucleic acids examined thus far, the molar ratios of total purines to total pyrimidines, and also of adenine to thymine and of guanine to cytosine, were not far from 1. As explained in the next section, this last observation would provide a crucial clue to Crick and Watson in elucidating the structure of DNA and in proposing a mechanism for DNA replication. And finally, Chargaff emphasized the fact that given the complexity now known concerning the composition of DNA, there will be no objection to the statement that, as far as chemical possibilities go, DNA could very well serve as one of the agents, or possibly as the agent, concerned with transmission of inherited properties. The first X-ray images of DNA were provided by Avery in 1936, and he noted that the molecule was capable of stretching 250% and that the spacing along the fiber axis is almost identical with that of a fully extended polypeptide chain system. However, he mistakenly suggested that its structure was composed of a close succession of flat or flattish nucleotides standing out perpendicularly to the long axis of the molecule to form a relatively rigid structure. And James Watson saw X-ray diffraction patterns of DNA for the first time in 1951 and was so excited that he moved from the US to England at the Cavendish Laboratory to be supervised by John Kendrew. It was there that Watson met Francis Crick for the first time and discovered their common interest in deciphering the structure of DNA, though this was neither student's official PhD project. At the same time, Rosalind Franklin was taking X-ray crystallography images of DNA in Maurice Wilkins' laboratory at King's College London. One day, Watson stopped by Wilkins' office for a visit, where he was shown an image that Franklin had recently taken. And as soon as Watson saw it, he was immediately convinced that DNA was a double helix. This was a novel idea since Pauling had suggested, in an article which had been provided to Watson and Crick in advance of publication, a three-stranded structure for DNA in which the phosphates are at the center of the helix with the bases on the outside, prompting Watson to comment that, "We were very struck by the ingenuity of the structure. The only doubt that I have is that I do not see what holds it together." Pauling's nucleic acid, in a sense, was not an acid at all. So Crick and Watson moved the sugar phosphate backbone to the outside of the structure as had previously been suggested by Franklin, which helped align the atomic distances in their model with the known crystallographic data. In addition, given the structure's twofold axis of symmetry, they realized that the two strands should run in opposite directions. The first model they produced had identical nucleotides paired between the two chains, but this required that the distance between the backbones varied to accommodate the different sizes of the nucleotides, since T and C are single-ringed molecules, whereas A and G are double-ringed molecules. So Watson tried pairing a single-ringed with a double-ringed nucleotide and found that this configuration was well stabilized by two hydrogen bonds between each base, and that any nucleotide could be inserted at any position in the chain without altering its physical properties, since the distance between nucleotides was now constant, and provided a solid explanation to Chargaff's observations that nucleic acids have equal ratios of A to T and C to G. When Watson showed Crick his model later that day, they knew they had solved the structure of DNA and told anyone who would listen that they had found the secret of life. On April 25, 1953, Crick and Watson's paper on the structure of DNA was published, and they noted that this model did not apply to RNA, as the extra oxygen atom would make too close a van der Waals contact. They concluded the manuscript by making a prescient observation that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. In a follow-up paper, they expanded on this last point, stating that if the sequence of one strand was known, the complementary strand could be written given the adenine only pairs with thymine and guanine with cytosine. In addition, they correctly suggested that, prior to duplication, the hydrogen bonds are broken and the two chains unwind and separate. Each chain then acts as a template for the formation onto itself of a new companion chain, so that eventually we shall have two pairs of chains where we only had one before. Moreover, the sequence of the pairs of bases will have been duplicated exactly. And so to summarize this video, DNA and RNA were found to be composed of nucleotides, molecules made up of phosphate, a pentose sugar, and a nitrogenous base, either a purine or a pyrimidine. In addition, thymine was shown to only be part of DNA, whereas uracil was only an RNA. RNA was shown to be mostly localized to the cytoplasm and was presumed to play a role in protein synthesis, whereas DNA was located to the nucleus, where it was presumed to be the genetic material responsible for heredity. Finally, Watson and Crick suggested that DNA was a double helix in which adenine paired with thymine and cytosine with guanine. Their suggestion fit well with the known crystallographic data, and their model suggested a mechanism by which DNA could be replicated. And so this brings this video to an end. I hope you enjoyed it. And if you did enjoy it and would like to know more about this topic, please check out my book, Landmark Experiments in Protein Science, using the link in the description. And if you have any questions, please leave them in the comments and I'll do my best to answer them. And I hope you'll join me for the next video on the way in which cells make energy in the form of ATP.